So it's a great pleasure to welcome you, Charlotte, to the Coronavirus Multi-Species Reading Group. I've been interested in your work for a long time, and it's lovely to finally have a chance to chat with you. I really want to get right into some of the interesting conceptual work that you're doing here. I think this notion of pluribiosis is a really interesting way to think about, the possible relations, ways of thinking about the relations that are emerging in a dynamic way between humans and their microbial clouds or swarms or whatever kind of way we want to think about it. You go into this idea of amphibiosis, which is a way, an earlier notion of thinking about microbes that might be good or bad in one situation and or another. But yeah, you're asking us to move beyond these sort of limiting dualisms to think about multiple potentialities in relation. So maybe if you could just tell us a little more about this concept, where it came from, and in the papers that you've offered, you've gestured to how it might be used in medicine. So maybe you could elaborate that a little bit.
Okay, actually at the beginning, I was a little, I don't know, I was not at ease with all the dualisms that we can cross in our different readings. I won't talk about nature, culture, biological, societal. It's not the point here. But I work with microbes for a very long time now, because I've made my PhD on the relationships between biologists and the yeast, Saccharomyces cerevisiae, in biological laboratory. So it was almost 15 years ago. And at that time, I already developed this relational way of thinking with microbes, and the idea that the laboratory itself is a space which is defined by the relationships between humans and non-humans. And so a space that has to be taken. I don't know how to say that. So you have to maintain this space. It's not something that is here, and it's not a rigid structure. It's something that is always transformed and dynamic. And so you have to respect a lot of rules regarding the organisms we work with in order to have this space maintained. And so after that, I worked also in clinical trials on HIV, and it was almost the same, how you keep viruses not under control, because it really is not what happens when you are an antiretroviral therapy, for example. It's just not a question of control. It can be thought of as a domestication process between the human, the virus, but also the entire society and the rules everybody had to put in place in order to live with and to becoming with this virus. So from the start, I was really not at ease with this idea of foes, friends. At that time, it was almost always foes, enemies, and these kind of things. And then I started to work on microbiota. And here it was like all of a sudden it was all about romanticized vision of our life with microbes, and the microbes are wonderful and blah, blah, blah. And it was not satisfying. And so I started to think about this question, and I'm clearly not the only one. There are a lot of people working on that, and I was really inspired by the work of John Dupré, Maureen O'Malley, Thomas Prado, and a lot of other people working on that questions. And so I was thinking about the spectrum, and I also worked with viral evolutionists who are always thinking not in terms of good, bad, enemies, or friend, but about spectrum of relationships. So that was the first step. And then I read the book of Martin Blaser, which is in so many ways criticable, but there was a very interesting concept in that, which was the concept of amphibiosis, which is a concept which was coined by Theodore, yes, I have the book here, Theodore Rosebery, in 1962. And this idea of the same entities which can have different relationships depending on the context or depending on the time, depending on the space, etc. And this was really interesting. And then the COVID-19 arrived, and I was a little bit, I don't know how to say that politely. Yes, I was hungry, actually, because a lot of people talked about viruses in ways that weren't very accurate, in my point of view. And so I tried to write a little paper, which is name is Politics of Amphibiosis, I think, something like that. Where I tried to explain that we have very ambiguous relationships with viruses, and yes, at some point they can hurt us, and actually COVID-19 did a lot and still do a lot of does a lot of bad things, but we also have mutualistic relationships with them or commensal relationships with them. And so I gave the famous example of syncytin, which are proteins necessary for placentation in mammals. And these, there are two syncytins, and they are the two of them are, both of them are coded by retroviral DNA or RNA, but transformed in DNA. And so at some point, viruses participate in our long history of evolution. And I know that Forrest talked about that also, but viruses play a lot of roles in immunity and this kind of thing. So I wanted to tell this story. And I published that paper, and I think that 2 days after that, I thought, yes, but no, it doesn't work neither, because yes, we have a spectrum of relationships, but the entities remain very fixed. And what we can see, and what is wonderful when you work with microbes, is that it moves all the time, and the entities are always transformed by the relationships that they are engaged in. And so I tried to find something to explain this just in one term, and because it's too long to explain that in each paper. So you have a spectrum of relationship, and then the entities are transformed and blah blah blah. And that's why I choose the term pluribiosis, which actually is a mix between Roman and Greek, but Latin and Greek. But anyway, that's not a big problem, I think. And so, yes, the idea of pluribiosis, there are different things now in that concept, but I'm still working on it. And so you have this idea of a descriptive thing. So relationships and entities, everybody are moving all the time, etc. But you also have this idea, then again, in that concept, there are so much people that I love. And so you have this idea of situated knowledges with the feminist epistemologies, and with Ahawe, with Barad, with everyone, all these wonderful researchers. And also the idea of situated biologies. So situated knowledges, situated by knowledge, I don't know, you don't say knowledges in English.
Yeah, knowledge is in the plural.
Yes, you can. Situated biologists. At the beginning, it was Margaret Locke who talked about local biologies, and then with Hans-Jörg Owner, I think, yes, I think it's in the paper they wrote together, they talked about situated biologies. And this is exactly what happens when you look at microbes. You see that, yes, you have big rules about biology, but each of one is instantiated in a different way, depending on the context. So yes, this is all that. And also the fact that, and that's the third aspect of pluribiosis, the fact that you have multiple knowledges, and so multiple ways of producing knowledge about the same kind of entities. Each of one will have consequences. And this, I think, is a wonderful, the best example of that is the wonderful paper of Hannah Landecker with the concept of involution that she uses to talk about the way the microbiology of yesterday is responsible of the biology of the bacteria of today. And so it also is a concept which contains, I don't know if we can say a moral thing, but at some point, the notion of pluribiosis is political because it stands against the idea of science as something which is natural and without consequences. And so it implies that we have to make some choices when we decide to use these knowledges in society.
Super interesting. Also want to give a shout out to Forrest, who's just joined, who's here with me in San Diego, now virtually, but later on today, physically. Hi, Forrest. Before you got on the line, she pulled your book off the shelf and was like, "Yay, the Bible! Here it is."
Yeah, the Bible is here.
So yeah, as per normal, if anyone has a question for Charlotte, feel free to just plunk it in the chat, unmute yourself, or put your hand up. And yeah, so I've got maybe another question that's kind of across the two papers, which is related to scalability. So you quote Anna Singh and talking about the ways that things might scale. And, these papers are making a strong argument against a one-size-fits-all approach to phage therapy. And part of what I'm wondering about in that context is just issues of maybe social justice and how we might think about, so for example, Forrest and I last night were talking about, some of the things I wrote about in my latest book with CRISPR, which is, and other kinds of gene therapies. Yeah, they're available, and leukemia, you don't have to go through intensive radiation or chemotherapy these days. You can get CAR T cells that are a really effective thing with very compared to chemo, like little side effects. But yeah, the price tag is $400,000 for a single dose, and then there's another gene therapy on the market, $2.1 million. And the kinds of intimate care work that you describe in the lab that produces a phage therapy for a particular patient, I mean, that's labor intensive, and you describe an infrastructure of antibiotics, but I'm wondering if we're going to make phage therapy available to people who aren't just kind of the elite, what does that look like if it's not a one-size-fits-all scalable model?
Yeah, that's all the question actually, because this is as I wrote, I think at the end of the paper, this is just the beginning at some point. But yes, I think that there are two, and Forrest will say if he agrees or not on that. But I think that there are two main dangers about using phage therapy as we used and continued to use antibiotics. The first is so you have two different ways now, two different models. You have the prêt-à-porter, so you are putting different phages, you are designing a cocktail in order to
slow down. You had two French phrases in the English article, and I didn't understand what they were. So
prêt-à-porter, ready to use.
Okay.
And sur mesure, so custom tailor-made.
Okay, so personalized medicine.
Yeah, but yes, but it's not totally. So I'm very cautious in using personalized medicine because it has a story. This concept has a story too, and I think it does not fit very well with phage therapy. But I still don't have another word. So yes, we can go with this one if you want. So there are these cocktails. So you put different phages in a bottle, and then you can use, for example, you can have different phages against Staphylococcus aureus, and you say, okay, so we designed that, we produce that in mass, and we can inject that to patients when they come to hospital, and we know, not for sure, but quite sure that they have a Staph aureus. And the other one is, okay, you go to the hospital, you have an infection. So by now you can't do that with people with acute infections, because when you have an acute infection, you have to go very fast. And so antibiotics are by far the best option. But when you are, and now we use these phages most of the time for chronic infections. So you have more time to find a solution. And then you will do a phagogram, as you can do an antibiogram, to see if the phages that you have in your bunk, so you need to have a bunk, and I come back to that later, but to see which phages are working. And then you can use two. Generally, the people I talk and I work with prefer to use at least two phages or three phages in order for the bacteria not to have the time to evolve and to find a way to resist a phage. So if you put one phage, two phage, and generally you put also an antibiotic, so normally the bacteria do not have the capacities to resist. That's on theory. And so, yes, by now, the majority of people I met are struggling to see the sur-mesure, the tailor-made approach to be developed, because we can see, so as I said, two problems. So the first is, if you use cocktails, and if the bacteria, if the strain of the patient is not, or if the phages are not really active on the bacteria, which renders the patient ill, then the bacteria could adapt and could develop resistances against those phages. And so it's not a good solution. So this is one problem. And then if it doesn't work, you also have to know that your body, and I think that Forrest said that in the multi-species reading group last time, your immune system reacts to phages. So it's not a one-shot treatment, but you have to be very careful, because if you use it once, probably the second time, it won't be enough efficient. And the second problem, I think, with phages, and if you start to use them as cocktails, is that if you use them in human health, then you could use them in animal health, and perhaps in environment, in agriculture, etc. And we literally have no idea of what would happen if we start to pour phages everywhere in the environment. We don't know. We have now a good view of what antibiotics did in the environment. So now the idea is really to be very careful with phage therapy. So we have to use it, and we have to use it well. We have to preserve antibiotics, and we also have to preserve phages. So regarding the model, in France now we have so I am really lucky, because we have a wonderful project which is in development in Lyon, where different people, it's an interdisciplinary team with microbiologists, pharmacists, infectious disease specialists, but also a public production factory. And so they joined together in order to be able to propose what I call an integrated model. So they can do everything, so they will do, we hope, everything from the isolation of phages from the environment to the administration to the patient. But there are a lot of work to do, because we have regulatory issues, we have political issues, we have economic issues. And we also, so France is not the only place in the world, and there are a lot of other things that happen in the same time. In United States, they have a wonderful initiative of hospitals working together in order to develop phage therapy, which is really good. But the really important thing to me, if we come back to social justice, is that at least in the French context, the only way to ensure that people will have a treatment not at a horrible cost is to go through the public system. But this needs strong commitment from the state, which is not something that is quite fashionable at those times. And so, yes, we are working in order to probably develop that. But just one point, and then I stop. Yes, the tailor-made approach is really important by now, but perhaps we don't know. Perhaps with the evolution of the knowledge and the evolution of the situation with AMR, perhaps we will need in, I don't know, 10, 20 or 30 years, phage cocktails. So we need another infrastructure, which is not the same as the antibiotic infrastructure, but we also need an infrastructure which is flexible in order to first be very careful in the use of phages. And then if it works and if there is no risk to democratize or to scale up the production and the use of phages, enable that.
Super interesting. Does anyone else want to jump in? I've got more questions, but definitely want to give other people an opportunity.
So I'll ask, so do you think we're going to get, I mean, yeah, I guess the hope is that we have like a couple thousand phage that everybody that have been tested and are ready to go. Is that what you think will happen? I mean, what's your vision of what it looks like in the 20-year life frame?
You want my wish, or you want
exactly your wish?
The pessimistic version. Honestly, I really don't know where we will be in 20 years. We have so much things to think about when we think about what the situation will be at that time. But actually, what I really impressed by the pace at which phage therapy develops now, because 3 years ago, the model in Lyon, I was wishing it, but I had no idea that others were working actively on it. And so I think that it is the beauty of phages, is that, you know, all these people, they do not want to, it was not their aim in life to design another model or another infrastructure for care. It's just that phages oblige them to do so, because they do not enter in the boxes, in the categories which were designed for chemical substances. So I don't know, perhaps it will be abandoned. Perhaps we will have new things, like we can use phage lysines, for example, or we can use antimicrobial peptides, or all these kind of things. I really don't know. And there are also other ways of using phages. You can also use phages to select bacteria sensible to antibiotic, and then put antibiotics in the IV, for example. It's so I really don't know, but what I really I think that I'm porté. I share the enthusiasm of the people I'm working with by now. So my view, in 20 years, we will have banks, phage banks, for each of the 6 most troublesome bacteria today. So the escape ones, so Escherichia, Klebsiella, Staphylococcus aureus, Acinetobacter bovanii, Pseudomonas aeruginosa, and all the Enterobacter species. And we will have these banks. We apparently, but I speak under a controlled forest, apparently it really depends on the bacterial species. But for example, you have very clonal bacteria like Staphylococcus aureus, and here we know that with few phages, we can have a good spectrum to cover a lot of different infections by Staphylococcus aureus, which is not the case, for example, for coli or for Klebsiella, which are not really even species. It's difficult to think about this kind of bacteria as the same species. But yes, the idea would be to have these collections of phages ready to use. So design it with the good manufacturing practices. And then to have, so now people are working on phagograms, which would be easy to perform in 6 or 8 hours, some even think about phagograms, which would be doable in 2 or 3 hours, but it's not now. It's in development, I think. So yes, you go to the hospital, and you can have these phages available. So it could be multicentric at some point. By now, for example, in Lyon, they really want to have a phage center, but perhaps in, I don't know, in 10 or 20 years, these collections will be duplicated in other hospitals and available in other places. And then you will be able to treat people with them. But it will never replace antibiotics. It totally is a different mode of action and a different mode of administration. And so you can't have the same thing as antibiotics. And at some point, I think it's the good thing, in order not to repeat the same mistakes we've made in the past.
One last question. Do you guys have like a good business model, good business people involved in it? Something that makes it sustainable, right? I mean, that's a real, it seems like that's the biggest challenge right now, or one of the biggest challenges.
Yeah. Actually, that's my part of the job, because so it's a kind of research action that I'm doing. At the beginning, I was just interviewing people and going to workshops and conferences and in labs and doing observation participation and this kind of thing. And at some point, I became to be really involved. And so now I'm part of this Lyon project. So we had just have a funding for 6 years, which is just the beginning. And my part is to, actually it's a double thing. So I follow the development of biomedical innovation. And at the same time, I try to work, not myself alone, but the idea is to work on a development model, which will ensure that it will be sustainable in time. And it's an economic problem, but it's also a regulatory problem, because by now phages are considered as drugs, and so they are under certain kind of regulation. So there's a lot of things that we can't do, and actually, that is in part why phage therapy is developing very slowly. So people are working, the Belgian team at the Queen Astrid Military Hospital worked for a long time now on these regulatory issues with Gilbert Verbeken and Jean-Paul Pirnet. And now in France, in Switzerland, also people are working on that. And the other part is, so you have this regulatory part and the economic part, which is to find a way to, for example, have the social security to reimburse this kind of treatment. How many does it cost, etc. The only thing I'm sure about is that there are also startups who are working on phage therapy, and I have, let's say that I see pretty well how many money they will ask for a treatment, and this is something that almost nobody can afford to. So, but yes, that's a crucial issue, and there are also the issue of patentability. Yeah. But again, another question.
Yeah. So I wonder that the only one that I know that did something like this was the Vertex model. And I don't know if you've looked at them, but they managed to come up with a business approach that allowed them to make drugs for cystic fibrosis, which is an orphan disease with a kind of the same problem, small number of people that need it. That the only advantage they have well, maybe that's true with phage therapy, that some of it is chronic, but usually you would think of it as being, acute. Yeah. Anyhow, I'll stop. Thanks. Good luck.
There's also the NDI. I think the NDI could also work. I also see that a lot of social scientists fellows are working on the question of antibiotic pipeline. So I'm thinking about Klaas Kirchele and I don't know how to pronounce it. I'm sorry. So there are. I think that this is something that can move. I hope so, actually. And also the fecal microbiota the FMT, the fecal microbiota transplant, which could be another model, because by now, for example, it's different because we know that poo is not something that costs a lot, but they developed models that, for example, in France, you can there is a center where they do FMT and they prepare this kind of, gélule, pills with poo inside. And so they sell it to other hospitals, but at a very low cost, which allows them to treat their own patients for free. And the cost is the same as a classical antibiotic, not a very expensive one. So that could be also another model.
That's super interesting. Thank you, Forrest, for those questions. It relates back to part of your paper where you're describing each phage bacteria pair is unique. So is the relationship between the technician and a given pair. So if you're talking about it in the context of fecal transplants, like you can think about there you're not only thinking about a technical process, but you're thinking about this vast organism, but also microbial multi-species assemblage. So there's a hand up from Rachel. That was more a comment, observation rather than a question. Rachel's got her hand up, but Zia had 2 questions in the chat before we get to Rachel. Do you want to ask those, Zia?
Sure, yeah. One of them, I was just curious about, in your paper, you mentioned resistance emerging from the kind of cocktail of phage therapy. And I wondered if you could just go into a little bit of the detail of that, if that was because there might be particular phages in there that certain bacteria were less vulnerable to, and therefore resistance would kind of develop and spread. So that was the first question. And the second one was about hearing a lot about phage therapy initially being kind of pioneered in Russia, or developed further in Russia than in the rest of the world. And I wondered if that meant that the kind of Russian phage science was much further ahead than the rest of the world, or maybe that initial start didn't give them too much of a kind of advantage in the science. I just wondering, yeah.
Perhaps I will start by the second question, and perhaps I will let Forrest answer to the first. Regarding the second, phages. So very quickly, phages were discovered. So there are a lot of different stories, and I remember, Forrest, actually, that we met in a bus on our way back to Bordeaux, and we were talking about Frederick Twert and Félix Derel. And there is a kind of controversy about who discovered the phages. Is it Twert? Is it Derel? And I really enjoy this story. But to keep it really short, so you had basically 3 people. You had Enkins in 1896, I think, that worked on the cholera bacillus and showed that there was something in the water of the Gange which was able to destroy this kind of bacillus, and this something was filterable in a Chamberlain filter, so it was something really small. And then you had a Twert who came back with the same kind of experiences, but also showed that this something was destroyed or inactivated by hot temperature. And he also added a lot of other things, but I won't go further in that. Then you had Félix Derel, and everybody talks about Derel because I think it was quite a guy. I'm not sure we would have enjoyed to work with him at that time. And he had a lot of stories, and it was awful at the Institut Pasteur. But at the Pasteur Institute, actually, he met with Georgi Eliava, which was Georgian student by the time, and they worked together, and they really enjoyed working together. So apparently, he was able to work with someone, and Georgi Eliava came back to Georgia and started to work on phages, and then he sent an invitation to Derel in 1933. And then Derel went to Georgia with his wife, and they together started to develop a kind of phage center. And then phage therapy was implemented, began at that time in Georgia. But also you have also people in Poland who are still working and very good working on phage therapy today. In Russia, you can buy phages directly over the counter in drugstores. And so I have the story of this patient. So I'm trying to writing a book now about all this. And I have this wonderful patient who suffers from urinary tract infection for a very long time because he's paraplegic. And he went to Russia and he bought phages in a pharmacy. And then he came back to France, asked doctors and the regulatory French agency to help himself. And they said, okay, no, we can't. It was produced in Russia. So we don't know the process of fabrication. We don't know anything about it. So we can't do that for you. So he did it all by himself. You can imagine he put himself directly the phages into his bladder. So it was a big thing. And so he's totally convinced by phage therapy. And so he's really, really angry against the French regulatory agency because they don't understand why this medicine, which is available in Russia and in other countries, is not available in France. And so I don't honestly about the kind of knowledge Georgian people have. I can't really answer because I wasn't able, because of the COVID-19, to go to BDC in the Eliava Center to do that work. I hope I will in the next couple of years. But the funny thing about Georgia and the rest of the world is that each time I go in a conference, in an international conference, and people say, oh, she's an anthropologist. So they came to me and said, okay, so you're an anthropologist. Can you explain to me why Georgians say that they know everything, but they won't tell us a thing? And I just have to remember them the story, of the relationships between North America and ex-USSR. And but honestly, it really is. It's weird, because even the most open people I know in France are have a kind of defiance with the Georgian people, knowledge, skills, know-how, etc. And so they say, yes, it's not really science, they put a little of this and they put a little of that, and we don't know how they do that, and blah, blah, blah. And when I meet patients who went to Georgia to be cured and who are actually cured, they say to me, wow, these French doctors, they are horrible because they said that Georgia, it's not science, but I'm cured now and I'm well and I still have my leg and I can walk and blah, blah, blah. So there's a very interesting field work to do in Georgia, for sure, but also on the relations and the tensions between Georgian and the North America and the Western Europe. Yes, I stop here, but it's a really interesting fieldwork to make. I really would like to do a ERC project and to have a lot of people working on that story, because by now I'm quite alone, and I would really enjoy to work with other social scientists. And I work with scientists and biologists and physicians, but not with social scientists. And that would be great.
Let's collaborate. Forrest, do you want to answer the first part of Zia's question, which was about cocktail phage therapy and resistance?
Well, I mean, it's honestly, I think you've done a great job of already talking about it, and that you have all the problems, right? Which are the evolution and the fact that the cocktails, I mean, people are finding where like one phage in the cocktail will inhibit another phage in the cocktail. So you get unfortunately, it's biology, and the phage have been having a phage on phage war forever. And so often you don't gain by having the cocktail. Sometimes you do gain by having the cocktail. Sometimes you gain by doing sequentially. Other times you don't. And that's really brings up the real challenge. I think it'll be exactly like what was proposed with the idea of like you're going to hit with a phage, and then you're going to hit with an antibiotic, and then you're going to hit with a phage again, or you know what I mean, something more like that than these cocktails of like 5 phage because of all the other complications that are popping up, roughly speaking. Yeah. And I'll quit there. The only thing I would say is also that tension between the scientists and the phage therapists is everywhere. It's not just between Georgia and the Western world. It's also within the Western world. Like there's really honestly separate meetings for people, and only in the last 10 years have you actually seen the scientists come together with the phage therapy people. Roughly speaking, yeah. It's a strong tension for various reasons.
Interesting. Rachel, go ahead.
Thanks so much for this. I'm learning a lot. I was just talking to some of my UCLA students about Catherine Waldby's work on tissue economies, and I guess you know, and actually the recent case reported in the news with Henrietta Lax's family members who have started a civil lawsuit over some of these patenting concerns and the appropriation without knowledge and consent of their mother's cell lining. And so I'm so curious, because you have this period in which you see a lot of maybe DIY biology going on, such as the example of, you know, people going across national boundaries and coming back with phage therapy opportunities. And then you also have your ethnographic work, you know, in laboratories and following the scientists themselves. And so I'm just so curious, as I'm thinking about, you know, this interesting moment where you're building a lot of new project possibilities there, where you are, and thinking about the kind of, as Forrest mentioned, the business model, and I guess my question is more to do with the archive that's being built. I mean, I think FMT is another example of this, you know, how the archive is being built for people who are interested in this therapy. And so I'm just wondering, I mean, you mentioned Blazer's work before, and I think perhaps one of the criticisms could certainly be questions about, you know, who gets to own this material, or colonial, you know, colonial concerns over the microbiome. And I just wonder what your sense is of, you know, where, you know, how building this archive might be happening with phage, and yeah, I don't, you know, what you've learned, or what you see, or what your question marks are. I'm just kind of leaving it open because I don't want to assume, but I know that's some of the debate in the broader kind of microbiome.
So that's really interesting, because yes, Catherine Waldby is on my desk right now, precisely because of the model, the development model question. Regarding the collections, for FMT, it's different. And a colleague of mine, Alexis Zimmer, is now working actively on the question of colonialism and the collections on FMT. So I won't talk about his work. But for phages, the interesting thing is that you can't you can't think about a phage alone. It's always a phage and a bacteria. So when you isolate a new phage, you isolate it on the bacteria. And the question, the interesting thing here is that to isolate new phages, you need bacterial bank. You know, you need banks of strains of a particular species of bacteria, for example, Staphylococcus aureus, or I don't know, Pseudomonas aeruginosa. And this kind of bacteria, you took them on patients when they went to hospital to be treated for their infection. And so these collections are, so I think it's something that I talk about in the other paper I sent about the microgeohistories. So these bacteria are themselves an archive. For example, the bacterial bank is an archive of the hospital and of the different patients who went to this hospital. You know, so you already have encapsulated in that collection a whole story of the different patients who came, when they came, why they came, the kind of strains they had, etc., the kind of treatment they had. And these banks are really well defined and characterized. So you have a lot of info about each strain, and it is really important because I won't enter the details because it's difficult, but you know, in bacteria you can have prophages, and the really thing you don't want when you produce new phages is to do that on the bacteria who has prophages in it, because it's for technical and scientific reasons. But so you have this first bank, and then you have the phages that you will isolate. So where do they come from? So they come from, generally, people use what they call rich waters. So waters from sewers or from lake or from rivers or whatever. But you know, it's a question I asked myself at the beginning of the work because of the Nagoya Protocol. So if you take some water in a country, do you have the right to go in another country and then to isolate phages from that water, which is not the water of your country? So I don't know how to put that, but so it's even more complicated because you have to think about two interrelated banks. It's not just you know, like the FMT or even I wrote when I was in PhD, I wrote a paper about the -80 freezer in the lab, which was the core of the lab and which was the archive of the laboratory because of all the strains of yeasts that were in it. But here it's all the more complicated because you have these two banks. And the other thing is that you have to freeze them, because if you don't freeze them, they continue to evolve. So that's another problem. And that's why perhaps the patentability issue won't be an issue at all, because how can you prove that your phage is the same as the phage in, for example, another collection? So it's a question of sequence homology. So your phage is moving all the time. So the phage in the hospital X won't be the phage in hospital Z, even if they are isolated on the same strain, because they will evolve differently in the process of isolation, purification, and characterization of the phage. So honestly, by now, I can't answer this question. I know that it is an issue, and it will be an issue in the next years. But yes, it really is complicated. And I really don't know. You know, the Henrietta Lacks example is quite good for that. But now the problem is a little bit different, because every patient now sign a paper when their bacteria is sampled or not. That is something that I should check. But yes, I'm sorry, it's a little, it's not very clear what I'm saying now, but yes, it's a very complicated issue, complex issue.
Thanks so much.
no one else has a question, I've got another one, and it's a very broad one. So in thinking about pluribiosis, you're thinking about the sorts of transformations involved to all the organisms involved in the encounter. So, you know, the paper's mostly about bacterial phage relations, but I'm wondering, as we're thinking about commensal relations, the possibility of symbiotic relations, if you're finding examples of the transformative potential of interspecific encounters that result in a lasting change in the human, or maybe not even lasting, but some sort of state change that you see as significant.
Yeah, now I can't give you an example, but I think there are plenty of examples of this kind. And also, if you consider that you don't put a strict boundary between what is biological and what is social. For example, when I wrote the first paper I wrote about pluribiosis was in French, and I gave the example of HIV because I think that for the better or for worse, HIV transformed the people who are infected by it, and for long term, and it also transformed the entire society. It transformed the way we interact with each other, the way we make love, the way we think about. And you have the same thing with COVID-19. For a time, the other was a potential source of danger. So yes, I think that I don't know to which extent we can use this concept of pluribiosis, but I think that it shouldn't be restrained to only biological changes, because, and actually, so the problem is that if we do that, it becomes something very classical, you know, it's just that relationship transforms anyone. It's just like, you know, I think we all agree on that. But examples, you have a lot of, so what is difficult humans are difficult to think with because of the temporality. You know, it's easy to think with microbes because during the lifetime of a human, we have a history, we have the impression of being the same person, and that can be contested, but anyway. But during this, I don't know, 20, 30, 40, 90 years, if we are lucky or not lucky, it depends. You have a lot of different generations of bacteria, viruses, fungi, and other creatures that evolve. And so I think that's the reason why I'm not comfortable with giving you examples about humans, is that they are very difficult to document. That's why the example with retroviruses is easier to understand, because yes, since it's encoded by ancient viruses, which integrating the genome of our ancestors at some point in evolution. And so we exist because of these viruses. And you have all the work of Lynn Margulis, which is plenty of this kind of symbiogenesis, you know, mitochondria, chloroplasts, and all these kind of things. So yes, I think that it's just a question of time and of how we apprehend, how we apprehend the different temporalities. Difficulty with humans is that we have to retrospectively reconstruct these kind of stories because we can't experience them in our flesh.
So Zia, go ahead.
So this might be a bit left field. So yeah. I was just thinking about the human virome and the ways in which, thinking in particular in relation to, let's say, gut ecology, and how phages maybe exist within the gut to regulate kind of gut health, and I'm sure in other places in the human body as well. And I wondered, I guess, two kind of related questions is one, have antivirals disrupted that? And is there a phage therapy that looks at less targeting, what's the word, recolonizing the human body of potential lost viromes, if that was an area of research that exists in relation to phage therapy. Does that make sense?
Yeah, actually, what is funny is that for the FMT, for example, so you use FMT to treat infections by a bacteria named Clostridium difficile. So FMT is efficient in 95 to 99% of the cases. So it's just like something that you don't have another example of such an efficient strategy or therapy. And people, it really is difficult because you take a sample, you take a whole ecosystem of a person and you put it in another person. And so you don't know, you have ideas, but you don't know, because it really is difficult to be sure of the causation mechanisms that are at play in this. But some people started to look at phages in these transplants, and they showed, so I don't want to say mistakes, but they showed that you have an increase of a certain kind of phages, but actually they are prophages, so they are, it's complicated, but they are not virulent phages, they are temperate phages, and apparently they play a role in the restoration of the health of the recipient of the transplant. And you can also, so there are a lot of different strategies by now. You can use FMT for decolonization, and you can use also phages. Perhaps not now, but some physicians want to use phages to decolonize some patients before, for example, surgery. So let's say that, for example, you have staph aureus in your nose, like 30% of people on the planet. It's not a problem, but if you have a surgery and if that staph comes to the wound, then you can have a nosocomial infection. And so some are thinking about decolonizing Staphylococcus aureus in the nose of patients before surgery, or in, houses for the elder ones. I don't know that you say that in English.
Nursing homes.
Mm-hmm.
Nursing homes?
Yes, nursing homes. So this is also a problem. So they think about, for example, decolonizing the nose of these people. I really don't know what to think about that. So it's not, I'm not promoting this kind of things. I just said that some people think about that. And I really don't know, perhaps there will be another step after that. And perhaps now phages is a new bacteria. So that's my point of view. So you can't think about bacteria without thinking about phages. It's so phages is a new black. But perhaps, I don't know, in 30 years, there will be another tiny creature, which will, it's like Russian dolls, so we don't know.
So with that, we are at the witching hour. Thank you for all the generative questions, and Charlotte for offering these fabulous papers, and for sharing your latest thoughts. So first of all, virtual clap.
Thank you. And thank you all for your questions.
And yeah, I do hope to continue the conversation. Next week on October 18th, 9 a.m. Pacific time, 6 p.m. Central European time, we have Jonathan Prekar, who's a PhD student here with another group in San Diego, and will be returning to conversations about COVID. He's got a paper that came out in Science a few months ago, looking at basically chains of interspecies transmission from animals to humans that basically fade out. And he's also been doing some work on revising the timeline of the pandemic emergence. So yeah, I hope to see some of you next week. And yeah, good to see you, Charlotte. See you soon, Forrest.
See you.
Thanks again for coming.
Thanks, Charlotte.
Thank you very much.
Bye, everybody.
Bye.